this session because if you look at the talk titles, it feels like a cellular couples therapy session. I want to advise you not to look for relationship advice here. And we'll start off with Aurora Nedelcu, who's going to tell us about a scandalous subject of cheating in cellular relationships.
I'm trying to find. Great, thank you so much. So I thought today, because it's the last day and we have a roundtable later on, I would give a talk that's a bit more conceptual rather than a lot of data. And also I'm hoping that some of the principles or ideas that I'm presenting might inspire some of you to look for analogous processes or genes in your systems. So with that, I'm going to just quickly give a quick overview of cooperation and cheating in the evolution of multicellularity, present you my approach and framework, and I'm going to actually spend a couple of seconds now on the goal and the take-home message, because now, warning, I tend to go to be late or no, to go long, so then in case I don't get to the take-home message, I want to have it up front. And then I'm going to introduce the model system and hopefully implications for cancer. When we talk about cooperation in terms of evolution of multicellularity, we can talk about the synergistic cooperation when everybody benefits, and this is something that now we are familiar as far as the undifferentiated multicellular groups. And again, there might be benefits, whatever those benefits are, depending on the group. What I want to talk about today is the costly cooperation or altruism, in which the behavior is costly to some members but beneficial to the group. And the best examples, of course, are somatic cells, which are now bona fide reproductive altruists, but also among altruists and among somatic cells, now we can also talk about some type of specialization. And again, with any type of cooperation, there's the temptation for cheating because it makes sense. Now, if you can still get the benefits of the cooperation without paying the cost, now the temptation is there, so to speak. So that, of course, is going to create a conflict between the interests of the cheater and the interests and the fitness of the group. If this is not controlled or mediated, the cheaters can increase in frequency because they have increased fitness, and that could actually result in the collapse of the group. And cancer cells are usually given as a best example of such cheaters. So in that respect, now we can think of cancer cells as somatic cell cheaters. Of course, that being said, then the question is, how do cooperative groups, including multicellular groups, mediate such conflicts and remain evolutionarily stable now over a long time? And there are many conceptual and theoretical frameworks I'm not going to touch on. Today I'm going to mostly talk about these issues from a mechanistic point of view. And again, mechanistically, one can imagine that you have to evolve mechanisms to suppress the emergence of cheaters. That might be suppressing genetic variations, suppressing the selective advantage of the cheaters. It might involve mechanisms to detect the cheaters or to police the cheaters. In medical or in evolution of multicellularity framework, such mechanisms are usually discussed as cancer suppression mechanisms. So cancer suppression mechanisms are just basically mechanisms that either suppress the emergence of cheaters or try to deal with them afterwards. Perfect. So again, today I'm going to talk about more of a mechanistic approach, like a bottom-up evolutionary approach, meaning that now I'm going to talk about the early evolution of multicellularity. And again, the idea being that cheating involves the breaking cooperation mechanisms that are involved in the group formation and somatic altruism. Today I'm going to talk about the somatic cell differentiation aspect of cooperation. The idea being that if we understand the basic principles that were involved in the emergence of such cooperative behaviors, then we might also understand how cheating is suppressed. In my lab or in my research for the last maybe 10 or so years, I'm obsessed with this concept of stress, and especially the possibility or the idea that the potential to evolve into more complex systems, whether it's a multicellular system or some other type of system, has to do to the ability to adaptively respond to stress. So the idea, again, if the environment now poses more perturbations, then you have to evolve more actions, meaning more traits, to be able to adapt. And this idea comes from this principle or law in cybernetics that basically states that in order to achieve complete control, the variety or diversity of actions a control system should be able to execute must be at least as great as the array of environmental perturbations that need to be compensated. And I like this cartoon because it basically says that now if you have 3 balls in the air, or 4, 2 hands might be okay. Now if you have more balls in the air, you might need more hands. So the idea that now environmental perturbations drive the number of actions that you have to evolve. Mechanistically, the hypothesis I've been trying to address for the last few years has to do with the possibility that mechanisms underlying cellular responses to environmental stress in unicellular organisms have been co-opted into the evolution of new traits, particularly cooperative traits like cell adhesion and differentiation, during the evolution of multicellularity. And from this follows that a loss of such traits would also come, in addition to now losing of the trait of the cooperative trait, would also involve the loss of the ability to respond to environmental stress. So I'm going to talk today about implications now of such loss for cooperation and for cancer. And I'm not going to talk about this today, but also I think that this idea can also have implication in terms of explaining why some systems now evolve more complex. There might be a correlation between changing or stressful environments in the sense of them selecting for more efficient stress responses. And if these responses can be co-opted into new traits, then one would expect that such environments to be more conducive to the evolution of complex biological systems. Oops, sorry. I have no idea how late I am already. So what I want to do today is to show evidence that the evolution of somatic cell differentiation involved the co-option of stress-induced pathways, and also to show you that actually this type of co-option provides a built-in system against cheaters, because cheaters are going to gain reproductive advantage, but they're going to lose in terms of survival in stressful environments. And I think that's true, that all organisms have to deal with stress in a way that suppresses their reproduction to increase survival, then probably similar mechanisms have been also involved in the evolution of multicellularity in other lineages, including animals, and that should actually reflect in cancer in terms of cancer cells should also be more sensitive to stress. So the model system, you already learned about this in the previous talks. The idea here is the Volvox has only 2 cell types. Somatic cells are specialized in survival. They are motile reproductive cells. They grow and reproduce. Each reproductive cell undergoes embryonic development to make a new baby that is similar to the parent. The genetic basis is very simple. There's only one gene that establishes the fate of the reproductive and the somatic cells. That gene is always off in the reproductive cells and always on in the somatic cells. So it establishes now the fate of the somatic cells. The somatic cells derive from the smallest cells at the end of embryogenesis. So REGA is considered a master developmental regulator. It's a transcription factor, contains a DNA binding domain of the type SAND. And again, this is an interesting story. It's only found in animals and green plants. And I kind of argue that maybe it was acquired by animals, by sponges, bilateral gene transfer. So I'm trying to get people with data on transcriptomics from sponges to look for this protein in their data, in their transcriptomes. Again, we don't know exactly why it gets expressed, other than that it's always expressed in the smallest cells. So we can call the REGA as being an altruism gene because now it's costly for the somatic cells, but beneficial for the group. Mutation as expected for altruism genes, mutations in this gene result in cheaters. So somatic cells start growing and proliferating. These are two different REC mutants. The only difference here is that these kind of tumor-like structures are in fact little babies. So the somatic cells regain not only immortality, but also totipotency. So if that's the case, you would expect a cheater like this actually to take over the wild populations, because now they have, each somatic cells can make a baby, so they have the reproductive advantage. However, we found that the mutants, green denotes dead cells, red is autofluorescence from chlorophyll. So the idea is that the mutants are very sensitive to light stress or heat stress. So although they have a reproductive advantage, they have an increased sensitivity to stress, and this can actually explain why we cannot find such mutants in the wild. So we consider this sensitivity to stress as a personal cost of cheating, since cheaters lose in terms of survival if they. So the next question, or an interesting question, is now how such altruistic genes can evolve. So the reggae gene is part of a gene family. There is a homology in Chlamydomonas, which is single cell and also non-differentiated volvocine algae. And we're wondering what such a gene can do in a single-cell organism. They don't have somatic cells. So I hypothesize that it's probably involved in the temporary suppression of reproduction to increase survival under stress, which is what we usually call an acclimation response. And indeed, the gene is expressed during nutrient stress in the stationary phase and coincides with the suppression of reproduction. If that's the case, then we would expect that the loss of RLS1 to increase the reproduction of the Chlamydomonas and decrease survival. And indeed, RLS1 mutants are not able to suppress, so this is the wild type, it's suppressing its reproduction in nutrient-deprived environments, as you expect. The mutants don't. In fact, the mutants under nutrient stress, the mutants grow much, grow better than the wild type, so they have the reproductive advantage that we predicted, but they are losing in terms of long-term survival. So basically, RLS1 appears to be a life history trade-off gene that increases survival under nutrient deprivation at the cost of reproduction. So this allows us to develop this conceptual framework where the co-option of a life history trade-off gene that is induced by environmental cues to increase survival at the cost of reproduction can be co-opted into a developmental gene such that it's expressed always on now in the soma and always off in the germ, and that allows to break this trade-off, because now in this context, both survival and reproduction of the multicellular organisms can be improved at the same time. So how can this happen? The working model that we have now is that the temporary stress can induce a signal, maybe a change in the redox status of the cell, that triggers the temporary proliferation and allows the Chlamydomonas now to survive under stress. So the idea is that the developmental cues, like the small cell size means an increased surface to volume ratio, for instance. So you expect some changes, intracellular changes in the small cells. If those changes in fact are the same changes that the ones induced by the temporary stress, then you expect that those signals would turn on the reggae, but now it's going to be permanently, because the cell is small all the time, and that actually induces the somatic cell differentiation. We also wanted to see if the reggae can still be induced environmentally, and indeed we can induce reggae both developmentally and environmentally. So reggae maintains its ancestral regulation, so it can be induced independent, both developmentally and environmentally, and this is important. And I also would like to now, at some point, I'm hoping that at some point other people will find such genes in animals, in early diverged animals, and if so, I expect that they evolve from environmentally induced genes in single-cell ancestors. So if this explains basically, if that's the case, then if you have a mutation in the reg, then you expect no proliferation, no somatic differentiation. The cell would grow, right? But they don't have the ability to acclimate, so they're going to try to proliferate, and they're going to proliferate, but they don't have the reggae to help them under stress, so they're going to die. So this explains why the reggae mutants are in fact more sensitive to stress. So the punchline here is that the reggae maintain its ancestral antagonistic effect on cell fitness. So yeah, RLS1 represses reproduction and increases survival in CLAMI, and it does the same in the somatic cell. So if you lose RLS1, no, you get the reverse, and that's exactly what you get in somatic cells, meaning that any cheater that would increase its reproduction, it's gonna come with a cost in survival. This idea of pleiotropy as a way to stabilize cooperation has been proposed a while back in the Dicty model system. What I'm suggesting is that co-opting stress responses could be actually a more general mechanism to stabilize cooperation, because all unicellular lineages know they have to be able to suppress their reproduction in response to stress. So if such mechanisms have been co-opted in other lineages into the somatic cell differentiation, that this co-option actually allows you to evolve now to have a built-in safety system for free. Because now if cheaters arose under this mechanism, then cheaters are going to have the advantage of reproduction, but it's going to always come with survival cost. So coming back to the second part, as I mentioned to you, I expect this is going to be the case also in other lineages, including animals, and that might explain, or this might also be a mechanism to suppress cancer cells early in their progression. So years back, I was in France in Frédéric Thomas' lab, my collaborator, and they told me about this hydra that they bring from the wild, and then they bring them in the lab. A few weeks later, they develop these tumor-like structures, and this is something that has been reported now from other labs. So I was curious why they cannot find the tumor of hydra in the environment. So I mentioned to them that one possibility is that when they bring them in the lab, they feed them every day, so they don't go through any type of stress. So I hypothesized that in the wild, precancerous or pre-tumoral cells might occur, but because of the changes in the environment, temperature between day and night, nutrient, and so on, and seasonal changes, those pre-tumoral cells might actually be purged out due to this mechanism that I mentioned. So based on that, they designed an experiment with both hydra and zebrafish. The zebrafish here has a it's a genetically engineered zebrafish to be able to induce melanoma at will. And they fed them 4 diets as far as the amount of food and the frequency. And as expected, they found that frequently overeating is a more risk. Actually, if they used a low-frequency feeding and a lean diet, they could inhibit completely the emergence of tumors. If they allow the tumors to arouse and then they started to the change in diet, then they could decrease or slow the progression of tumors. But you can prevent actually the tumor emergence by playing with the amount of food and the frequency of food. So basically, and by the way, and this is actually consistent with previous studies with cancer cell lines in vitro that showed, for instance, the starvation, that cancer cells are most sensitive to starvation. Just a couple of papers in case you want to look at. So to conclude about cancer, so my hypothesis is actually precancerous cells, and I'm talking about precancerous cells before they accumulate a lot of mutation that allow them to adapt to various microenvironments. Precancerous cells are more sensitive to stress because of this trade-off between reproduction and survival. So precancerous cells, they proliferate, right? They have mutations in proliferation mechanisms, and those mutations are likely having an antagonistic pleiotropic effect on survival, so they're going to make them more sensitive to stress. So because of that, I think many of the precancerous cells are purged out from our body before they get to acquire additional mutations. And this sensitivity to stress of the precancerous cells is something that now we inherited from our ancestors. And this could be actually, in fact, an early anti-cancer mechanism that we inherited. So if that's the case, one might ask, now, why do we still get cancer? And the answer might actually have to do with what in evolutionary medicine we call mismatch with the current environment. Our ancestors actually were going through now periods of starvation, they were experiencing a lot more perturbation in their environment. We actually now, with our eating habits, eating all the time, so basically the precancerous cells are not exposed to the type of perturbations that would expose this sensitivity to stress. So I still think that this evolutionary vulnerability of cancer cells can be exposed and exploited. So the take-home message again is that now if when it comes to cooperation, if mechanisms that have been co-opted for corporations are in fact mechanisms involved in mechanisms that were beneficial at the individual level, like personal benefits, breaking now those mechanisms are going to come with a personal cost. So usually I make this kind of analogy that now if when I cooperate, what I'm using my hands, and then if I want to break out, if I cut here, right, I'm losing also my ability to eat. So I'm not only getting out of the cooperative, I'm also losing something personal. So this type of cost can stabilize cooperation. So I am expecting that the co-option of such stress response is a more general mechanism. And with that, thank you very much.